Hello boys, welcome back to another session of online biology video tutorials. This session is meant for class 12th biology students. This is for 7th July 2021. As all of us know, we have been discussing about chapter number 3, human reproduction. In our previous sessions, we discussed about male reproductive system, significance of each part of it, female reproductive system, significance of each part of it. We also thoroughly discussed, deliberated the physiological, anatomical, biochemistry part of each structure. In this session, we are going to deal with what is called gametogenesis. As the name suggests, Genesis means taking birth. Gametes, reproductive cells. Don't get confused between gonads and gametes. Gonads are organs. Okay? Like testis is a gonad. Sperm is a gamete. Testis is a male gonad. Sperm is a male gamete. I hope you are now clear the difference between gonad and gamete. Gonad is reproductive organ. Gamete is reproductive cell. Similarly, in a female gonad is ovary and female gamete is ovum. Now in this session we are going to discuss about how sperms are produced, male gametogenesis, and how ova or ovum is produced from ovary, female gametogenesis. Going ahead, we talk about this. As we said, it is the formation of reproductive cells or sex cells from reproductive organs or sex organs. Here, the male gamete is the sperm, female gamete is the ovum. Male gonad is a testis, female gonad is ovary. Now, types of gametogenesis, two types as I said. One is the formation of male gamete, that is sperm, spermatogenesis. Genesis, taking birth, creation. What is to be produced, what is to be created? Sperm. So, spermatogenesis. Similarly, oogenesis, birth of ovum. Oogenesis, formation of ovum. For this, of course, you need to practice this diagram, that is the longitudinal section of ovary. Already we have discussed this when we were talking about the female reproductive system, and this is important because even in practicals, you need to identify this under a microscope, longitudinal section of ovary, draw the diagram and label it. That is for 3 marks. There is a slide, okay? Identification, slide identification. So you need to first start with the primary follicle, label primary follicle. What is the significance of it? What becomes primary follicle? Identify that. Second, primary follicle grows and becomes matured, forms a secondary follicle. So what is it? What is the significance of it? Secondary follicle becomes matured and forms Graafian follicle. What is the significance of that? Graafian follicle on maturity breaks off and releases ovum or secondary oocyte. What is the importance of that? What happens to the left out material, that follicular tissues? They degenerate and form corpus luteum. What is the significance of corpus luteum? So once you identify the significance of each part of it, of course there is blood flow, arteries and veins. Then you get the concept of how ovum is formed in a female. Moving ahead, we talk of first the male reproductive cell formation or sperm formation, spermatogenesis. So it is the process of formation of sperms. Technically, sperms are called spermatozoa in seminiferous tubules of testis. Remember, 2 days back we talked about the anatomy of male reproductive system. In that we identified in the TS of testis, seminiferous tubules, right? So how from seminiferous, or how sperms are formed in seminiferous tubules? That's what we are going to discuss under spermatogenesis. First, formation of spermatids. There should be first formation of spermatids, and this is followed by Spermiogenesis. Now don't get confused again, Spermatogenesis and Spermiogenesis. Spermatogenesis is formation of sperms, male reproductive cells. Spermiogenesis is transformation of spermatids into sperm. What is this transformation? There will be change in the shape, there will be change in the size, there will be change in biochemistry part of it, anatomical structure. All these transformations are transfers, changes together, we call it transformation. And finally, a spermatid becomes a sperm. This process of spermatid becoming a sperm is spermiogenesis. So, formation of spermatids, sperm mother cells. Remember we talked about PMC, pollen mother cell, in second chapter, sexual reproduction in flowers. And then we talked about MMC, megaspore mother cell. Like that here, sperm mother cell, SMC. What are these SMC? They are basically spermatogonia. They are mother cells that undergo meiosis to form sperms. So they produce spermatids basically. And the spermatids transform into sperms. Now under spermatogenesis, what happens? In the cross section of the seminiferous tubules you can see here, formation. First, spermatogonia, they are diploid, 2N, two sets of chromosomes will be there. The spermatogonia basically present in testis, they are testicular cells. They undergo mitosis, so they develop the numbers, form thousands of primary spermatocytes, and they maintain the chromosome number. Diploidicity is maintained. Now, primary spermatocytes, this one, primary spermatocytes undergo meiotic 1 or meiosis 1. And as all of you know, meiosis 1 is RD, reduction division. What is reduced? 2N is reduced to N. That is, diploidicity is reduced to haploidicity. Two sets of chromosomes have been reduced to N. In particular, since we are talking of human reproduction, the cell, each cell present in the testis will be having two sets of 23 chromosomes. One set is 23. So like that, two sets, 23 into 2. So N is equal to 23, hence 2N. This gets reduced into just one set. Only 23 chromosomes will be there. Okay. Since the testicular cells will be having 46 chromosomes, and in this 46 chromosomes, 44 are autosomes and 2 are sex chromosomes. As all of us know, in any male, Y chromosome will be present. So it is the 2 sex chromosomes or allosomes will be very invariably XY. So when this gets reduced from this 44 autosomes, 50% of it, 22 autosomes will come. And out of the X and Y, one sperm will get X, another sperm will get Y. So that means there is variety in the sperms. So this 23, as we said, N number, and then this undergoes meiotic 2. Okay, so primary spermatocytes undergoing meiosis 1, which is reduction division, will form secondary spermatocyte. Secondary spermatocyte undergoing meiotic 2, which is, we say, ED, equal division, will form spermatids. Now, what we said, spermatids which are haploid will undergo transformation, and this transformation leads to formation of spermatozoa, or in common language we say sperms. Now, you know, when the spermatids undergo transformation leading to change in the shape and size, you see, basically these are spherical spermatids, When they form spermatozoa, look at the change in the shape, change in the size, change in the anatomy, change in the physiology, and change in the morphology. So we can see the spermiogenesis taking place. That is what we said. Transformation of spermatids to spermatozoa is spermiogenesis. But remember, in this number is maintained. Haploidicity is maintained. So one sperm may be having 22 plus X, another sperm may be having 22 plus Y. Now this decides exactly whether male baby is going to be taking birth or female baby is going to be taking birth. That means it is the sperm. I am very careful with my terminology. It is the sperm that will decide the sex of the baby. I am not saying it is the male or it is the husband. It is the sperm. If it is sperm carrying X, 22 plus X, fuses with ovum 22 plus X, 44 plus XX, female baby. If it is the sperm carrying 22 plus Y, fuses with the ovum 22 plus X, 44 XY, male baby. And in this, all of us must understand that the female has no choice in this, because ovum will have only one set of chromosomes, 22 plus X. So why I am saying this, in our society many a time the blame is put squarely on a lady, on the mother when she delivers a female baby. And you know, female feticide. Many babies, female babies, are getting terminated even before they take birth. And the female is blamed for that. And many diverse cases, all this is happening. So the purpose is that there is absolutely no mistake on the female side. There is no need to put blame on the mother. After all, mother is a mother. Whereas, I am not saying that we should put blame on the father, no, or the male. Because the father or the male doesn't have physical choice of selecting a sperm carrying 22 plus X or 22 plus Y. So what decides? It is the chance factor. Okay. Either of the sperms can fuse. And there are conditions we will discuss before, in fact, as we go ahead. So spermatogenesis is the formation of sperm from testicular cells. And when we take the vertical section of the seminiferous tubule, we can see the various cells, interstitial cells, then you have got spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and they get transformed and form spermatozoa. This is how you have to, again, in this slide, you have to identify under a microscope, draw the diagram and identify the label. Maybe when the external examiner asks you the significance of each one, you should be in a position to explain to him both spermatogenesis as well as spermiogenesis. So keep on practicing diagrams. That is what, being a biology boy, you must draw beautiful diagrams, scientific diagrams of course. Label it, and as I have been telling you, labeling should be arrow mark towards the part like this, arrow mark towards the part, arrow mark towards the part. And then understand the significance of first origin, spermatogonia, from where it is coming. Okay, testicular cell. What is the importance of this? Without spermatogonia, there is no formation of primary spermatocyte. So like this you must analyze. And then when you label Serotonin cells, what is the significance of these Serotonin cells? Okay, so then you get the complete concept just by drawing the diagram and labeling it. So primary spermatocyte leads to secondary spermatocytes. Secondary spermatocyte leads to formation of spermatids. Thus, one primary spermatocyte gives rise to formation of 4 sperms. So, since it is meiosis, you know, at the end of meiosis, one mother cell will produce 4 sperms. And the spermatogenesis, when we see under electron microscope, the sperms will be appearing in this way. So after spermiogenesis, sperm heads are embedded in Serotonin cells to get nourishment. So here you come to know what is the significance of Serotonin cells. They are meant for providing nourishment to sperms. Then they are released to lumen of seminiferous tubules. That's why these seminiferous tubules will be filled with sperms. It is called spermiation. Look at this, the spermiation process here. And continue with that. Overall, is there any influence of brain? It's all happening, in testis, okay? But is there any control over this formation? Is there any role of the brain? Definitely. Without the involvement of brain, nothing happens in our body, as all of you know. And basically, the brain controls the release of hormones. And what are hormones? Hormones are biochemical substances. They play heavy role, huge role in that. So basically, hypothalamus present in the brain secretes a hormone called GnRH. What is this GnRH? Gonadotropic releasing hormone, GnRH. And this gonadotropic releasing hormone acts on the pituitary. There is a gland called pituitary gland in the brain. And immediately the pituitary secretes gonadotropins. Okay? It secretes two gonadotropic hormones. What are those? One is the LH, luteinizing hormone. Another is FSH, follicular stimulating hormone. Now the luteinizing hormone acts on Leydig cells. There is presence of Leydig cells in the testis. And immediately the Leydig cells under the influence of LH secretes male sex hormones called androgens. Like we said, testosterone. And this androgen secretion stimulates the formation of sperms. Spermatogenesis will be stimulated. And what about FSH, follicular stimulating hormones? They act on serotonin cells, which are meant for nutrition to the growing sperms, and they stimulate secretion of factors for spermiogenesis. Because we said spermiogenesis means transformation. How does the transformation take place without the involvement of brain? Nothing can be changed in our body without the involvement of brain. So it is the brain which is the boss for us, for everything that happens in our body. So unless until the brain releases certain factors, spermiogenesis will not take place. This is the major role of our human brain on the reproductive system. And the spermatogenesis, once the sperm is formed, the sperm will be like this. It has got three parts: head region, middle piece, and tail portion. And the head region has got what we call plasma membrane covering it. There is a short head region at the anterior acrosome, which helps in penetration, and nucleus all deciding. It's more powerful one. There is a neck region, and most important in the middle piece is number of mitochondria will be present. And mitochondria are the powerhouses of the cell. They give power to the sperm. And of course, tail helps in locomotion, movement, swimming. So basically, a mature sperm is about 0.06 millimeter length. Sperm is enveloped by plasma membrane. It has three regions: head, middle piece, tail. You can see end piece is 5 micro, and principal piece, middle piece, head piece, total together it is just 0.06 millimeters. And the head region coming to, it's oval shaped as you are seeing, formed of nucleus and acrosome. It consists of two parts. Acrosome is formed from Golgi complex. It contains lytic, means breaking enzymes. That is the importance of acrosome. And behind the head is the presence of neck. And in the middle piece, it is composed of axial filament, okay, surrounded by mitochondria and cytoplasm. And mitochondria produce energy for the sperm motility. Here, motility of the sperm is the deciding factor during fertilization. That means more the mitochondria, more the energy for the sperm, more the energy for the sperm, higher the speed, higher the motility. Chances of getting selected for fusing with the ovum is high. And the last portion, tail, it consists of central axial filament. The sperm moves in fluid medium and the female genital tract by undulating movement of the tail. That is the importance of tail. Without tail, there won't be movement, and the sperm without movement is almost a dead one. The human male ejaculates about 200 to 300 million sperms during a coitus. During copulation, the sperm will be released, and there are two. The fastest will be the winner, and survival of the fittest. For normal fertility, at least 60% of the sperms must have normal shape, as I said, and size. They should not lose the shape and size during the journey. And then 40% of them must have vigorous motility. That means they must be 100-meter dash. They must be champions. So the fastest one will be qualified for this. Out of how many? 200 to 300 millions. How many sperms finally selected? Only one. See the competition. You talk of competition in UPSC, NDA. You talk of competition in NTSC. You talk of competition in NEET. What about here? Compared to that, this is great. 1 out of 300 million sperms will be selected. Hence, that is the competition. In our next class, we will talk about the form